Welcome, fellow knowledge explorers, to the Deep Dive.
Great to be here.
Today, we're plunging into a topic that often lurks, beneath the surface of our awareness. But its impact is profoundly shaping the health of our planet. Marine diseases.
Yeah, it's something most people don't think about much.
Exactly. You might typically think of epidemics as, well, terrestrial concerns, right?
Yep.
Affecting humans, farm animals, or crops.
Mm-hmm.
But what happens when disease outbreaks unfold in the vast, fluid world beneath the waves? It's a realm that operates by entirely different rules.
Absolutely. Fundamentally different.
So our mission today is to unpack a fascinating stack of sources. We've got comprehensive reviews on marine disease ecology, some specific deep dives into devastating outbreaks affecting really iconic species.
Right.
We're going to explore how marine diseases are fundamentally different from their terrestrial counterparts, what truly drives their emergence and spread.
And the links back to broader ocean health.
Precisely. And the surprising, often critical, insights they offer about the overall health of our shared planet. We'll uncover why the ocean's health is so intrinsically linked to our own.
It really is all connected.
Okay, so let's dive in. It's easy to overlook, I think, but parasites and disease-causing organisms, they've always been an integral part of natural systems, haven't they?
Oh, absolutely. They shape ecosystems, they drive evolution. They're not inherently bad, they're just part of the natural order.
Right. But for a long time, their significance in marine environments has been, well, the sources say, comparatively unknown.
That's true, especially when you contrast it with how much we've studied and recognized the importance of parasites in terrestrial ecosystems, on land.
So why that historical oversight? What do you think led to that, and what have we potentially missed by not looking closer underwater sooner?
Well, that's a really fundamental question. And what's truly fascinating, or maybe concerning, is that despite this historical oversight, marine disease-causing organisms are having significant impacts.
Right, not just minor things.
No. Significant impacts on marine species and entire communities. This isn't just a minor ecological footnote. It's a real driving force that shapes populations, it influences species interactions, and it can even restructure entire ecosystems.
Wow.
For decades, I think our focus on disease was, understandably, very anthropocentric, driven by the immediate needs of human public health, agriculture, managing livestock.
Things that affect us directly.
Exactly.
Yeah.
We developed these really robust theoretical frameworks and practical methods for understanding epidemics on land, and that naturally became the default lens through which we viewed all disease.
Makes sense.
But the ocean, think about its immense scale, its opacity. You can't just easily see what's going on down there, and the sheer, almost bewildering diversity of life within it. It remained, in many ways, a kind of scientific black box.
Yeah.
Trying to study microscopic pathogens in a vast three-dimensional water column, subject to dynamic currents, unseen depths. It's just inherently far more challenging than, say, monitoring a cattle herd in a pasture or observing a crop field.
The logistics alone must be a nightmare.
Immense. The logistical and technological hurdles were, and honestly, in some cases still are, huge. And that really delayed our understanding of the ocean's unique epidemiological landscape.
So we're playing catch-up now.
In many ways, yes.
Yeah.
And we've seen stark demonstrations of these significant impacts and the profound consequences of maybe not looking sooner, particularly in recent decades.
Like what kind of demonstrations?
Well, there have been numerous disease outbreaks causing mass mortalities across an incredibly wide range of marine taxa. Think about the sheer breadth of this.
Right.
We're talking about everything from charismatic megafauna like the harbor seals in Europe. Remember those devastating die-offs from Facinus temper virus?
I do, yeah. Tragic.
And Florida manatees, where harmful algal blooms producing brev toxins have taken such a severe toll, leading to widespread sickness and death.
Like the red tides and things like that.
Exactly. But the impact extends far beyond these, easily recognizable charismatic animals. It reaches down to the fundamental ecosystem builders and engineers.
Like coral.
Like corals, definitely. But also things like coral and algae, which are vital for building reef structures. Vast kelp forests, people call them the rainforests of the ocean for a reason. Crucial seagrass beds, which act as nurseries and store carbon.
So the very foundations.
The absolute foundations.
Yeah.
And of course the corals themselves, constantly battling various syndromes, bleaching events. It's a tough time for corals.
It really is.
And beyond those, we've seen widespread impacts on other invertebrates like scallops and abalone, impacting fisheries, diverse fishes, like the Australasian pilchard, where viral outbreaks just decimated populations over huge areas. The pervasive nature of these threats across such a wide spectrum of marine life, it really underscores how widespread and serious the problem of marine disease has become. It's not just isolated incidents.
And is it getting worse? I mean, the sources mention a trend.
Yeah, the data strongly suggests a troubling and pervasive trend. For some really important marine taxa, diseases and their impacts appear to have increased over the past 30 years or so. 30 years?
Yeah.
That's relatively recent in ecological terms.
It is. And this isn't just anecdotal. It's a statistically significant pattern we see in specific groups. Marine turtles, for instance, where diseases like fibropapillomatosis we'll talk more about that have become a major conservation concern.
Okay.
Corals facing a multitude of diseases, white band, black band, stony coral tissue loss disease, often made worse by warming oceans.
Right.
Various mollusks impacting shellfish aquaculture. Sea urchins, which have suffered these devastating die-offs in places, and perhaps most strikingly, marine mammals, increasingly susceptible to morbilliviruses and other pathogens.
And this increase, it's been noted since around 1970.
Particularly noted since 1970, yeah, suggesting a kind of acceleration more recently. And this isn't just one or two hotspots, it's a widespread and growing concern. It paints a broader picture of escalating vulnerability across marine ecosystems.
So the very fabric of marine life seems to be under increasing pressure from disease.
That's a good way to put it. And it's prompting urgent scientific inquiry into the underlying causes. Why is this happening now?
That's a crucial point then, that the ocean really plays by very different rules when it comes to disease. Our conventional understanding, epidemiology, as you said, it's largely driven by protecting us, humans, our crops, our farm animals.
Right, the terrestrial focus.
So most of our theories, our management methods, they're rooted deeply in medical or veterinary examples. But the ocean, as you said, is a fundamentally different beast.
Completely.
So what does this all truly mean for applying these established terrestrial theories to marine environments? Are we just trying to force a square peg into a round hole here? Or can we adapt our terrestrial playbook?
That's an excellent way to frame it. And the answer is, well, it's complicated. Yes, the basic processes modeled in epidemiology, how a pathogen spreads, infects, impacts a host population, those are fundamentally universal principles.
Okay, so the basics still apply.
The basics, like infection rates, recovery rates, transmission efficiencies, they remain constant in their abstract form. However, and this is a big however, there are major qualitative differences between marine and terrestrial environments.
Right.
And these differences have substantial implications for applying standard epidemic theory to the ocean. It really is like trying to apply the rules of chess to a game of Go.
Okay, I like that analogy.
Some fundamental concepts like strategy apply, but the board, the pieces, the objectives are entirely distinct. We absolutely need to rethink how these basic processes manifest and interact underwater. The physics and biology of the ocean fundamentally alter disease dynamics.
So what's the first big difference?
Well, one striking and often overlooked difference is just the sheer biological richness, the higher taxonomic diversity of marine environments.
Higher diversity, how so?
Consider this. On land, you find representatives from only 9 of the 34 recognized major animal groups of phyla.
Only 9.
Wow.
Only 9. That means nearly 3/4 of all animal phyla are just absent from land. But in the sea, all 34 phyla are present, thriving in various forms.
All of them?
All of them. This incredible disparity means marine ecosystems offer a far greater taxonomic, life history, and functional diversity for both hosts and parasites.
So way more players in the game.
Way more players. It creates a much more complex host-parasite evolutionary milieu. This dynamic, intricate web of coevolutionary relationships unfolding for hundreds of millions of years in a vastly more diverse genetic landscape.
Okay.
And what's more, even among the phyla found in both habitats, more classes of organisms are involved in parasitic relationships in the sea than on land.
So even within the shared groups, there's more parasitism.
Exactly. It translates to an exponentially larger array of potential host-parasite interactions, each with its unique dynamics. It makes it incredibly challenging to generalize or predict outcomes based solely on terrestrial models. The sheer number of possible interactions is just unparalleled.
Okay, that's a huge difference. What else?
Then there are profound differences in life histories. How organisms live, reproduce, disperse.
All right.
Marine organisms often have life histories that diverge significantly from terrestrial ones. For many animal hosts, especially sessile or benthic invertebrates, think corals, sponges, many mollusks. A key distinction is that their dispersal stages are usually microscopic eggs or larvae.
Tiny little things drifting in the water.
Exactly. Released into the water column, drifting with currents. On land, dispersal stages are typically adult or subadult individuals actively moving, potentially carrying a pathogen directly from A to B.
Right, like a migrating bird carrying bird flu.
Precisely. This fundamental difference means that host dispersal stages in the ocean, those tiny larvae are often less likely to be infected by the same pathogens that affect the adult.
Why is that?
Well, imagine a tiny, free-swimming coral larva, maybe just millimeters long. It's vastly different in form and function from a large, complex, sessile adult coral colony. The pathogens adapted to infect the adult's complex tissues might simply not be able to infect or even survive in that tiny pelagic larval form. Or the larva's immune system might handle it differently.
Okay, so the disease might not travel with the babies.
Often, yes. This radically shifts disease dynamics. The primary way a host population spreads geographically might actually be largely pathogen-free. This impacts how diseases persist, how they reemerge in new locations, or how long they can survive during dispersal. The disease often has to kind of wait for a susceptible adult form to establish itself in a new spot.
That's fascinating. What else is fundamentally different?
Perhaps one of the most crucial differences for disease spread is more open nature of recruitment in marine environments.
Open recruitment, what does that mean?
Terrestrial models often assume relatively closed populations. Individuals are largely born, live, and die within a defined area, with limited movement in and out.
Okay, like a herd of deer in a specific valley.
Right. But marine populations are typically highly connected. Recruitment, the process where new individuals enter a population, often comes from external sources. Larvae are carried in by currents over vast distances from distant larval pools.
So populations aren't isolated.
Not nearly as much. This potential for long-distance dispersal of larvae, or even infected mobile adults, can enable unusually rapid propagation of epidemics in marine systems. It's like the ocean currents act as superhighways.
Highways for disease.
You got it. We've seen this with that herpes virus ravaging pilchard populations in the Southern Ocean. It spread over massive areas, multiple countries. And morbillivirus infections in marine mammals, spreading widely, sometimes even against prevailing currents because the infected animals are moving.
Wow, even against the current.
Yeah, it shows the power of oceanographic connectivity. Now, this openness can also buffer local host populations from the immediate devastating impacts of an outbreak, because there's a continuous influx of new, potentially uninfected individuals from elsewhere. Ah, so it can help recovery, too.
Yeah, it can. But it's a double-edged sword. It simultaneously facilitates the persistence of local diseases over broader geographic areas by constantly reintroducing pathogens. It keeps the fire smoldering, so to speak.
Okay, that makes sense. You also mentioned something about unique life forms, modular colonial ones.
Yes, that's another key difference. A unique characteristic of marine environments is the prevalence of modular colonial life forms, like sponges, corals, bryozoans, ascidians.
Things that grow like building blocks.
Exactly. Organisms composed of repeated, often genetically identical building blocks derived from asexual reproduction. They often form these large, sprawling colonies covering huge areas of the seafloor.
And we don't have those on land.
Not in the animal kingdom, no. They're completely absent in terrestrial animal environments. Plants fill somewhat similar foundational, sessile, often clonal role on land, but it's different.
So why does this matter for disease? Functionally, it matters immensely, because the genetic homogeneity, the fact that they're often clones, can facilitate the buildup of more virulent strains of pathogens.
Ah, the monoculture idea again.
Precisely. It's a fundamental difference. Imagine trying to stop a disease in a field where every single plant is genetically identical. If one gets sick, they all have the exact same vulnerabilities. The pathogen can just sweep through.
Right.
Couple that with the generally rapid evolution of pathogens compared to their much slower evolving, often long-lived hosts, well, it really facilitates the epidemic spread of virulent pathogens. It's like a natural monoculture, inherently more vulnerable.
And being stuck in one place doesn't help either, I guess.
Exactly. Their sessile nature also makes them more apparent and predictable in space. A pathogen doesn't have to search for its next host as much. They're just there, waiting. This can lead to higher infection rates compared to mobile hosts. It's a key reason we see such rapid and devastating coral disease outbreaks, for example.
Okay, so life history, connectivity, colonial forms.
Yeah.
What about the parasites themselves? Are they different too?
They are. The types of parasites also vary significantly. Think about parasitoids, parasites that kill their host as a necessary part of their development, like that classic parasitic wasp larva eating a caterpillar from the inside out.
Right, common on land.
Very common, especially among insects. But they're relatively uncommon in the ocean. Instead, something called parasitic castrators are remarkably common in the marine environment.
Parasitic castrators, what do they do?
They completely prevent host reproduction. They effectively sterilize the host, but they don't normally kill it.
So the host lives, but can't reproduce.
Exactly. And while that might seem less dramatic than being eaten from the inside out, the impact is insidious and highly significant ecologically. Infected hosts remain in the population. They keep eating, taking up space, potentially competing with uninfected reproductive individuals.
Even though they're reproductive dead ends.
Right. This can have really weird long-term population consequences. Models suggest that a sterilizing microparasite that doesn't affect mortality can actually have a greater effect on its host population long-term than one that only increases mortality.
How so?
Because it tends to destabilize host dynamics. It can lead to cyclical behavior, boom and bust cycles driven by the disease prevalence. It's a subtle but profound difference in how disease impacts population dynamics underwater.
Fascinating. Okay, and finally, what about how diseases actually get from one host to another? Transmission modes.
Right, transmission modes also vary considerably. Let's look at a few key ones. Vertical transmission parent to offspring.
Like HIV in humans.
Exactly, important in many terrestrial systems, but it's likely to be less important in many marine organisms with complex life histories, especially those with tiny eggs or larvae.
Because the parasite can't easily infect that different life stage.
It seems difficult. The physiological hurdles for a pathogen to bridge that huge developmental gap from a complex adult to a simple larva seem substantial. However, there's a critical exception. Mutualists, like those photosynthetic algae, zooxanthellae, living inside corals, they are often vertically transmitted, passed down from parent coral to offspring larvae.
Ah, okay.
So it suggests that maybe the rarity of known vertical transmission in marine pathogens might just reflect an absence of evidence. Maybe we just haven't looked hard enough or developed the right detection methods yet. It's possible it's happening more than we realize.
Okay, fair point. What about vectors? Like mosquitoes for malaria?
Vector diseases transmitted by mobile organisms moving parasites between hosts, they appear to be less common in marine than in terrestrial environments. Again, maybe due to insufficient study or the challenges of the marine realm.
They do exist.
Oh yes, some have been identified, like blood parasites of fishes that use leeches and these little crustaceans called gnanthaid isopods as transporters, much like ticks or mosquitoes on land.
Right.
And more recently, it's been shown that fireworms can actually spread an infectious Vibrio bacterium among corals, acting as a biological vector.
Fireworms spreading bacteria.
Wow. Yeah. But generally, marine systems seem to have lower searching efficiency for vectors, maybe because of the vastness and fluidity making encounters less predictable. And there seem to be fewer true blood-borne pathogens that would absolutely require a blood-feeding vector.
Okay. And the last one, pathogens just surviving out in the environment.
Right, the survival of free-living pathogens. Whether infective stages generally survive longer in marine or terrestrial environments, honestly, it's not clear. It's highly context-dependent.
Depend on the bug in the environment.
Exactly. Terrestrial bacteria typically don't survive well in marine environments. The salinity, the osmotic pressure, it's usually lethal for them.
Okay.
But conversely, some native marine and estuarine bacteria, including several human pathogens, particularly species within the genus Vibrio, which we'll definitely come back to, they can survive for surprisingly long periods in the marine environment, just hanging out in the water or sediment, maintaining their infectivity.
So the ocean itself can be a reservoir.
A persistent environmental reservoir. And that has huge implications for both marine life and human health, because these reservoirs can serve as continuous sources of infection, making disease control far more difficult.
We've just unpacked how the very fabric of marine life really changes the rules for disease. But to truly delve into these unique challenges, it helps to speak the same language, using precise terms. Our sources lay out some crucial definitions.
Absolutely. Terminology is key. Let's clarify some core concepts, especially since this field borrows from both medicine and ecology, sometimes with subtle shifts in meaning.
Okay. First up, disease itself.
Right. Broadly, disease means a condition that impairs normal function. It doesn't have to be caused by a transmissible agent, though often, especially in ecology, we use it as shorthand for infectious disease.
Got it. And epidemic.
An epidemic refers to a rapid increase, peak, and then a decline of a parasite or pathogen outbreak in a population. The word comes from Greek meaning upon the people, but we use it for outbreaks in any animal or plant population. You might also hear epizootic, which specifically means an epidemic in an animal population. They're often used interchangeably in ecology.
Okay. Then you mentioned different parasite types, macro and micro.
Yes. Macroparasites are usually multicellular thinkworms like helminths or arthropods. When we model their impact, we often consider the actual number, the burden of parasites per host.
Counting the worms, basically.
Pretty much. In contrast, microparasites are typically unicellular microorganisms, viruses, bacteria, protozoans. For these, modeling is often simplified by classifying hosts into discrete states: susceptible, infected, or resistant or covered. We don't usually need to count individual virus particles.
Makes sense. And that modular colonial life form.
Right. That describes organisms like corals, sponges, bryozoans, composed of repeated, often genetically identical units or modules derived from asexual reproduction, forming a larger colony, unique to marine animals.
And open population.
An open population is one where most new individuals, the recruitment, come from outside sources carried in by currents rather than being born solely within that local population. Very common in the sea.
Okay. How about just parasite?
A parasite lives in an intimate and durable association with usually one host individual of another species per life stage, and it has a detrimental effect on that host. It benefits at the host's expense.
And those subtypes, parasitoids and castrators.
Right. Parasitoids kill their hosts as a necessary part of their development, consuming them less common in marine animals. Parasitic castrators prevent host reproduction without typically killing the host, letting the infected individual persist but remain sterile.
Got it. Pathogen.
A pathogen is specifically a disease-causing microorganism that multiplies within its host. Its effects are generally controlled or fought by the host's immune response.
Okay. And R0. Sounds mathematical.
It is. R0, R0, is the basic reproductive number. It quantifies the average number of secondary cases generated by a single primary infected case when a pathogen is first introduced into a completely susceptible population.
So how fast it spreads initially?
Exactly.
Yeah.
If R0 is greater than 1, the disease will spread, potentially causing an epidemic. If it's less than 1, it'll likely die out on its own. It's a key indicator of transmissibility.
Crucial number. Vector.
A vector is a mobile organism that transmits a parasite or pathogen from one host to another. The mosquito, the leech, the fireworm we mentioned.
Right. Vertical transmission.
Parent to offspring. Direct transfer during reproduction or early development.
And virulence.
Virulence is the degree of harm caused by a parasite or pathogen to an infected host. How sick it makes them, how much it reduces their fitness or lifespan. Importantly, don't confuse virulence with transmissibility, R0. A very virulent pathogen might kill its host too quickly to spread effectively.
Good distinction. And finally, zooxanthellae.
Those are the single-celled photosynthetic algae that live symbiotically inside many marine invertebrates, most famously corals. They are crucial mutualists, and as we noted, often vertically transmitted.
Understanding these terms is definitely crucial because, as you've made really clear, the ocean plays by some very different rules when it comes to disease. These distinctions frame how we look at marine health.
Exactly. They're the foundation for understanding the specific cases we're about to discuss.
Okay, let's take all that theory now and apply it to a very real, very widespread marine disease. Fibropapillomatosis, or FP, in sea turtles.
Right, a classic, well-studied example.
It's a neoplastic disease, you said, meaning abnormal tissue growth, essentially tumors. Benign, usually, but still devastating.
And it's affecting sea turtles across the globe.
That's right. FP is characterized by the development of these single or multiple epithelial or fibroepithelial tumors.
What does that mean, fibroepithelial? It just refers to the cell types involved both fibrous connective tissue and the surface skin layer, the epithelium. Pathologists identify them specifically as papillomas, fibropapillomas, or fibromas based on the microscopic structure.
And these tumors, they can appear anywhere.
Literally anywhere on the turtle's body. From their sensitive eyelids and beaks, which obviously impairs feeding and vision, to their powerful flippers, impeding swimming and migration, even on their shells and soft tissue. And in advanced stages, and this is where it gets really insidious, they can develop internal tumors.
Internal ones, too.
Yes, often far more life-threatening than the visible external ones. They can affect vital organs, lungs, kidneys, intestines, potentially leading to organ failure.
Wow. How long has this been known about?
Its history goes back Quite a way. The first formal report was in 1937 in free-ranging green turtles, Chelonia mitis, in Florida, though it was likely around before then, just maybe not documented scientifically. By the 1980s, we started seeing cases in captive green turtles, too, raising concerns about transmission in controlled environments. And then, alarmingly, from 1990 onwards, the prevalence just shot up.
Increased dramatically.
Alarmingly, yeah. Especially in well-studied areas like Florida and Hawaii, it went from being a relatively rare thing to a major health crisis for those green turtle populations, really impacting conservation efforts.
But you mentioned it declined in Hawaii later.
Interestingly, yes. It later declined over time in Hawaii, for reasons that are still not entirely clear, which itself is an area of active research. It highlights the complex, often unpredictable dynamics of these diseases. Why did it drop off there? We don't fully know.
Huh. So it's not always a one-way street?
Not always. Currently, FP has been reported in all species of sea turtles.
All 7?
All 7 existing species, yeah. From the huge leatherback to the little Kemp's Ridley, and in practically all the world's oceans. That global distribution is really striking.
What does that suggest? Very widespread cause.
It points to either a very pervasive causative agent or perhaps widely distributed environmental cofactors that trigger the disease, or likely a combination of both.
Right.
But the incidence varies a lot. By species, by specific population over time, and depending on the location and environmental conditions. It's not uniform.
And can turtles survive it? You mentioned regression.
They can. While FP can certainly have fatal consequences, leading directly to death through organ failure or starvation if tumors block the mouth, it's also really important to note that regression of tumors has been reported.
So they can shrink or disappear?
In some cases, yes. The tumors can shrink and even disappear completely, which offers a glimmer of hope for recovery. Though again, the precise mechanisms behind this spontaneous regression aren't fully understood. Why do some turtles recover and others don't? That's a critical question for ongoing research.
Okay. Let's talk about what it looks like. You said tumors can be anywhere, but what's typical?
Affected turtles typically present with 1 to, well, sometimes dozens of these fibroepithelial tumors. And size range is huge, from tiny, like 0.1 centimeters, barely visible,
Really?
to truly massive growths, exceeding 30 centimeters in diameter, sometimes as large as a dinner plate on a big turtle.
Good grief.
And the larger ones frequently show necrosis tissue death and ulceration, open sores. That makes them highly susceptible to secondary bacterial or fungal infections, which further weakens the turtle.
Okay. And what do they look like? Texture and color?
Appearance varies widely. They can be firm or smooth. Color ranges from pale pink, whitish, yellow, all the way to darker greens, purples, browns, grays, even black. It depends on location, blood supply, secondary infections. This variability can make diagnosis tricky just by looking, which is why pathology is important for confirmation.
And the impact goes beyond just the tumors themselves, right?
Oh, absolutely. Severely affected turtles are often emaciated, really thin and wasted away. They're weak, lack energy. They're often anemic, low red blood cells, leading to fatigue. And critically, they're immunosuppressed.
Their immune system is weak.
Severely compromised. This makes them highly susceptible to heavy loads of other parasites and increases their risk of developing secondary infections from bacteria or fungi that wouldn't normally harm a healthy turtle.
That's a downward spiral.
It really can be. Plus, large external tumors, especially around the eyes or mouth, cause severe functional problems. Blindness, buoyancy issues, making it hard to dive or surface.
Can't feed properly?
Can't feed, kidney failure, gastrointestinal blockage preventing digestion, and in some cases, just direct death from organ failure or starvation.
And affects growth, too.
Yeah, it's been reported to decrease growth rates in afflicted turtles, making them less competitive, more vulnerable, and it has negative effects both at the individual level, survival, reproduction, and at the population level, impacting reproductive success and overall viability. That's why it's such a major conservation concern for endangered sea turtle species.
Okay, so what causes such a devastating disease? You mentioned the scientific community thinks it's multifactorial. Right. Multifactorial etiology, meaning it's likely not just one single thing, but a combination of factors acting together, a kind of perfect storm scenario.
But there's a prime suspect, right? A virus.
There is. FP has been associated with several viruses over the years, papilloma-like viruses, retroviruses, things known to cause growths in other animals.
Yeah.
But the one that has systematically been detected in FP tumors.
Consistently found.
Consistently found, yes. And even sometimes in clinically healthy turtles across multiple studies, different oceans, is the Chelonid herpes virus 5, CHHV5, also known as scudavirus colonid alpha 5.
A herpes virus, like cold sores in humans.
It's in the same broad family, the alpha herpes viridae, a common group of double-stranded DNA viruses causing various diseases in vertebrates. What makes CHV5 the most likely culprit? The etiological agent.
The cause.
The cause, yes, is its consistent detection right in the tumor cells, and the fact that scientists have managed to get it to replicate in vitro in green turtle skin cells in the lab.
So they can grow it in the lab. They can get it to replicate in cells? Yes. That's a strong indicator it can actively infect and multiply in the host cells. It demonstrates its pathogenic potential, a crucial step in proving causality.
But there's a but, isn't there? The sources say it's still inconclusive.
There is a but, a significant one. Despite this really strong circumstantial evidence, definitive confirmation of CHV5 as the primary causal agent of FP tumors remains technically inconclusive.
Why? If it's always there and can replicate.
It comes down to Koch's postulates, those strict criteria scientists use to definitively link a specific microbe to a specific disease. Remember Robert Koch?
Vaguely from biology class. 4 steps.
4 steps. 1, find the microbe in all diseased animals, not healthy ones. 2, isolate it and grow it in pure culture. 3, infect a healthy animal with the culture and cause the same disease. 4, re-isolate the same microbe from the newly diseased animal.
Okay, a rigorous checklist.
Extremely rigorous. And for CHV5, despite decades of effort by dedicated scientists, there's been this persistent, frustrating difficulty in cultivating and isolating the virus sufficiently to satisfy all those postulates. Especially step 3, reliably causing the full disease in healthy turtles under controlled conditions, and then completing step 4.
This is like having the suspect's DNA everywhere, but not being able to get the final confession or replicate the crime perfectly.
That's a pretty good analogy. We have a mountain of evidence pointing to CHV5. It's almost certainly involved, likely the primary agent. But getting that final definitive proof, according to those very strict 19th century rules, has been the elusive piece. It remains an ongoing scientific challenge.
That really highlights the difficulties in marine disease research.
Okay.
Let's narrow our focus now to a specific region, northwestern Mexico, Baja California Peninsula, parts of the mainland, bordering the Pacific and the Gulf of California.
Right, a critical area for sea turtles.
Super important habitats for feeding, development, reproduction for 5 of the 7 sea turtle species. So, what does the data tell us about FP there? Is it a big problem? Or is it still largely unknown?
Well, a recent scoping review looking at data from 1990 to 2024, it actually reveals what the authors call an important empty gap of knowledge concerning FP in this crucial region.
A gap in knowledge, so not much data.
Historically, relatively little published data. Only 32 marine turtles with FP and/or herpes virus infection have been formally reported in the scientific literature for NW Mexico within that 34-year timeframe.
32 isn't many over that period for such a large area.
It isn't. And of those, only 14 were confirmed cases. Using robust diagnostic techniques like histopathology, looking at tissues under a microscope, transmission electron microscopy, TM for ultrafine detail, and molecular methods like PCR to detect viral DNA.
Okay, solid evidence for those 14.
Right. The remaining 18 were only suspected cases, based solely on visual inspection of external lesions without any confirmatory lab testing. So, less scientifically rigorous, could potentially be misdiagnoses.
So the data scarcity itself is a key finding.
Exactly. It points to a real need for more systematic, standardized monitoring and diagnostic efforts across this vast and ecologically vital region. We probably don't have the full picture yet.
When were the first confirmed cases actually reported there?
Relatively recently, in 2014. In olive ridley and loggerhead turtles from Baja California Sur.
Okay.
But what's intriguing is that the herpes virus itself, CHV5, was actually detected in clinically healthy black turtles, turtles that looked fine way back in 2004, also in Baja California Sur.
Healthy turtles carrying the virus.
Apparently. This suggests the virus might have been present, perhaps in a latent or non-symptomatic state, within the population for quite some time before visible tumors became more common, or at least more frequently detected and reported.
And has it been increasing recently?
There's been a notable recent trend, a rapid increase in reports of FP tumor presence between 2019 and 2024.
An actual increase in the disease?
Possibly. But the review is careful to note that it's also highly probable this reflects an increase in monitoring effort and better recognition and understanding of the disease by local scientists and conservationists.
The looking harder effect.
Exactly. It's always a challenge to interpret disease trends when surveillance efforts improve over time. Are we seeing more disease, or are we just getting better at finding it? It's likely a bit of both.
Where are the cases concentrated geographically?
They're not uniformly spread out. 24 of the 32 reported cases, that's 75%, were found in Baja California Sur State, BCS, including specific well-known turtle hotspots like Magdalena Bay, Ojo de Libre Lagoon, and the Gulf of Ulloa.
Okay.
And notably, 9 of these cases were reported from El Suspiro Beach, which is a solitary nesting beach for olive ridley turtles. That's significant because FP reports in olive ridleys often came from mass nesting Arabata beaches further south.
Interesting. Any cases elsewhere?
Yes, other confirmed or suspected cases were found further east in Sonora, San Pedro Island, Quino Bay, and Sinaloa, Navatuste.
But none in the northern Baja state or Nayarit.
No FP or CHV5 reports were found in the states of Baja California, BC, or Nayarit within the review period. However, the review explicitly states this apparent absence seems linked to a lack of systematic monitoring in those areas rather than proof the disease isn't there.
Absence of evidence isn't evidence of absence.
Precisely. It just highlights where future research needs to focus.
What about the species affected? Mostly green turtles, like elsewhere.
Black turtles, which are the regional subpopulation green turtles, represented 50% of the total reported cases, and mostly immature individuals, which fits the global pattern. Juvenile green turtles often use these coastal feeding sites more heavily.
Okay.
Olive ridley turtles were next, representing 46.8% of cases. Mostly adult females, many found on those nesting beaches, which is a notable finding for solitary nesters in this region. And finally, just one immature loggerhead turtle represented 3.2% of the cases, so less common, but still present as a host there. This species breakdown gives us clues about susceptibility and habitat use.
What about the tumors themselves in an NW Mexico? Are they as bad as in places like Florida?
Well, based on the available data where lesion size was measured, that was for 89 lesions from 24 turtles. They typically ranged from 0.2 up to 11 centimeters in diameter.
11 centimeters, still pretty big.
It is, but the review notes this size range is relatively low compared to cases reported in Brazil, where you can get up to 129 tumors per turtle, or long-standing hotspots like Hawaii and Florida, where tumors often exceed 10 centimeters, sometimes reaching 30 centimeters.
Why the difference? Maybe the disease is newer there.
That's one possibility. Perhaps the disease hasn't been present as long, so tumors haven't had as much time to grow really large or proliferate extensively on individual turtles.
Where on the body do they mostly appear?
The anterior or front portion of the body was affected in about 85% of cases. The anterior flippers were the most common site, around 46%, followed by the neck, around 22%, probably because there's more exposed soft tissue there.
And what do they look like typically?
Most had a nodular, kind of bumpy and verrucous or wart-like appearance with a rough surface, often described as looking like cauliflower.
Cauliflower.
Yeah.
Okay, were there any really severe cases found?
Yes. One adult black turtle found in Ojo de Libre Lagoon was severely affected. It had multiple coalescent meaning merging together and ulcerated cauliflower-like tumors larger than 10 centimeters. So severe, life-threatening cases definitely occur in the region, consistent with global patterns.
And did the tissue analysis, the histopathology, show anything unique?
The analysis consistently showed characteristics seen globally with FP, that abnormal proliferation of skin layers, the epidermis and dermis, and degenerative changes within the cells. Nothing fundamentally different there. But interestingly, one clinically healthy loggerhead turtle one that looked fine externally also showed histopathological changes consistent with FP. And critically, it had intranuclear inclusion bodies, or IIBs.
Inclusion bodies, what are those?
They're microscopic structures inside the cell nucleus that are indicators of active viral multiplication, what's called lytic replication. The virus is actively reproducing within the host cells.
Even though the turtle looked healthy.
Exactly. Even without any visible external tumors. This is a really critical finding.
Why is this so critical?
Because it suggests that different species, whether they look healthy or are visibly sick, might be actively shedding or releasing viral particles into these common shared feeding areas, even if they don't show outward signs of the disease.
So healthy-looking turtles could still be spreading the virus?
Potentially, yes.
Huh.
This has profound implications for understanding how the virus transmits in these shared marine habitats. Apparently, healthy individuals could still be acting as vectors or reservoirs for the disease.
Okay, that complicates things. Now, connecting this back to the bigger picture, the emergence and spread of FP raises that critical question. How do human activities influence these dynamics?
Right, the environmental factors.
We're talking about things beyond just the virus. This sounds like that core principle you mentioned earlier, the One Health approach.
Yeah.
Can you explain how that applies here?
You've hit on a crucial connection. This is precisely where the One Health approach becomes so vital. It's a transdisciplinary, collaborative way of thinking that recognizes the health of oceans, marine organisms, and humans are all intrinsically influenced and interconnected.
Everything affects everything else.
In essence, yes, it's a holistic view.
Yeah.
You can't truly understand or manage the health of one piece of this vast system like sea turtles without considering its interactions with all the others, including us humans and our impacts on the environment, from local pollution to global climate patterns.
So how does this apply to FP? How are human activities suspected cofactors?
Well, scientists strongly suspect that human-driven activities act as crucial cofactors in the disease's development and severity. It's not just about the virus being present, it's about environmental conditions, largely altered by us, creating a perfect storm that allows the virus to manifest as these debilitating tumors.
Okay, like what specific conditions?
One significant link is with eutrophication.
Nutrient pollution, right?
Exactly. Often caused by nutrient runoff from things like agricultural fertilizers, untreated sewage, urban discharges flowing into coastal waters. Higher FP occurrence has been reported in highly urbanized areas with eutrophication.
And how does that affect the turtles or the virus?
The thinking is that eutrophication leads to a decrease in the diversity of macroalgae, the seaweeds that green turtles eat. Invasive algae species often outcompete native ones in these nutrient-rich, polluted waters.
Okay.
And the macroalgae that thrive in these impacted environments are often invasive species which have a greater potential to sequester high amounts of nitrogen, N, in the form of arginine.
Arginine, the amino acid.
Yes. And arginine has been reported as a potential tumor promoter in other biological systems. So the implication is that the quality of the green turtle's diet, which is directly modified by human activities causing eutrophication, could be a key cofactor. It might directly impact their susceptibility to tumor growth and progression.
Wow. So what we put into the ocean literally changes their food in a way that might promote tumors.
That seems to be a strong possibility. It's a profound link between our waste streams and wildlife disease.
Are there other environmental links? Biotoxins?
Yes, other studies also report a potential relationship with various biotoxins, things like Limbia toxin A, Brev toxins, okadaic acid.
Produced by algal blooms.
Exactly.
Okay.
Produced by harmful algal blooms, toxic dinoflagellates, cyanobacteria that proliferate on algae and seagrasses in these nutrient-enriched waters. Turtles then consume these toxins along with their food.
And the toxins could contribute to FP.
It's hypothesized they could, either by directly promoting abnormal cell growth or, perhaps more likely, by weakening the turtles' immune systems, making them more vulnerable to the effects of the herpes virus. The exact mechanisms are still being investigated, but the correlation is definitely concerning.
What about pollutants, like pesticides or heavy metals?
That's another area of suspicion. While no direct causal link between FP and things like organochlorine pesticides, OCPs, PAHs, polycyclic aromatic hydrocarbons from oil spills combustion, or heavy metals has been definitively established as the primary cause.
Okay.
It's been widely proposed that chronic exposure to these compounds could cause immunosuppression in sea turtles.
Weakening the immune system again.
Right. A weakened immune system, compromised by persistent exposure to pollutants, could play a significant role in tumor progression, especially in areas impacted by mining, industrial waste, or heavy agriculture.
And NW Mexico, despite seeming pristine in parts, does have those activities.
It does. While often perceived as relatively pristine, NW Mexico is also one of the most important agro-industrial regions in Mexico. It experiences significant urban discharges, agricultural runoff, and mining activities in certain areas.
So turtles there could be exposed.
Potentially, yes. For instance, one study in Novagist, a site where FP cases were reported, showed that frequent consumption of local snappers by humans could pose carcinogenic risks due to OCP accumulation in the fish. That tells you those compounds are in the food web.
Right.
Similarly, high concentrations of metals like iron, copper, cadmium, magnesium have been found in macroalgae and seagrasses in key feeding grounds like Magdalena Bay and parts of Sonora.
Stuff the turtles eat.
The stuff the turtles eat. These potentially toxic substances could be incorporated into their diet, posing a chronic health risk and potentially predisposing them to diseases like FP.
So putting it all together.
These findings collectively reflect the exposure of marine life to a wide range of polluting sources in NW Mexico's coastal ecosystems. And these are strongly suspected as environmental triggers for FP manifestation and severity.
which makes turtles sentinels.
Exactly. Sea turtles are widely considered sentinel species. Their health responds visibly and profoundly to environmental changes. They act as living indicators for coastal environmental quality.
So the increase in FP there could be a warning sign.
It very well might be. The suspected increase in FT incidence in NW Mexico's feeding sites could serve to alert us about possible negative impacts on the health of the entire coastal ecosystem, and in turn, maybe even potential negative effects on public health through things like contaminated seafood or just general ecosystem degradation. The interconnectedness couldn't be clearer.
So what this all means for us, it's a powerful reminder that the health of a marine turtle, or any species really, isn't isolated. It's like a mirror reflecting the health of its entire environment, and by extension, our own.
Absolutely.
When you consider how much our actions are influencing these complex systems, what really stands out to you about our responsibility in shaping the health of these ancient mariners and the ecosystems they depend on? It's a profound connection to think about.
It really is. It forces us to confront the consequences of our activities, even those far inland, on the health of the ocean.
Okay, from the more subtle, complex impacts of FP in turtles, let's shift gears now to something even more dramatic, almost apocalyptic sounding. Sea star wasting syndrome, or SSWS.
Yeah, SSWS was and is a major event.
This isn't just about individual animals dying. The sources say it's about the very foundation of coastal ecosystems. So first off, why are sea stars so important? What makes them foundational?
Well, sea stars, or starfish, as many people call them, they're far more than just pretty creatures you see in tide pools or on the seabed. They are indispensable keystone predators within many coastal marine ecosystems.
Keystone species, that term again, meaning disproportionately important.
Exactly. A species that has a huge effect on its environment relative to its actual abundance. You remove a keystone species, and the whole ecosystem can fundamentally change, sometimes even collapse.
And sea stars are like that. Which ones in particular?
Iconic species like the sunflower sea star, Pycnopodia helianthoids, that's the really big one, up to 24 arms, move surprisingly fast.
Wow, 24 arms.
Yeah, impressive animal. And the ochre star, Pisaster ochresis, that's the common, often brightly colored purple or orange one you see clinging to rocks in the intertidal zone. These guys play a critical role in stabilizing complex food webs.
How? By eating things.
Primarily, yes. Through their voracious feeding habits, they act as top-down regulators controlling populations of other invertebrates, most notably sea urchins.
Ah, they eat sea urchins.
They love sea urchins, and by keeping urchin populations in check, they prevent the urchins from mowing down entire kelp forests. Without sea stars, urchin populations can just explode.
Okay, so they protect the kelp forests indirectly.
Critically important for kelp forests, so the crisis that emerged back in June 2013, it was truly unprecedented in its scale and severity.
What happened in 2013?
SSWS, sea star wasting syndrome, began as this sort of poorly characterized nonspecific syndrome. The initial symptoms were a bit vague, could have been mistaken for other things. Okay.
But it very quickly culminated in these massive mortality events across numerous sea star species all along the Pacific coast of North America. Geographically, it stretched from Baja California in Mexico all the way up to the chilly waters of Alaska.
The entire coast?
Pretty much the entire west coast of North America. This event has been formally recognized as the largest known marine wildlife epizootic epidemic in animals ever documented.
The largest ever. That's staggering.
The sheer scale is hard to comprehend. Conservative estimates suggest over 5 billion sea stars were lost across that vast range.
5 billion.
Billion, with a B. And populations of the sunflower sea star, that big important predator, experienced a precipitous decline of up to 90% across its entire range.
90% gone.
Basically wiped out in many areas. This wasn't just a regional decline, it was an ecological catastrophe affecting a foundational group of organisms. It signaled a profound disturbance to these coastal ecosystems. And the symptoms. The sources describe them as gruesome. It sounds like a rapid, horrifying decline. Can you paint a clearer picture?
Yeah.
What actually happens to them?
Yeah, gruesome is unfortunately accurate. The syndrome manifests as this complex pathological process that progresses with really alarming speed.
Okay.
Initial observable symptoms often include a sort of deflated appearance. The sea star loses its normal turgor, its plumpness. It looks sunken, flaccid, not firm and robust.
Like it's losing water pressure.
Kinda, yeah. Often accompanied by abnormal postures. You might see them twisting their arms upwards or into rigid, unnatural positions, or they just seem unable to hold on to rocks.
Losing control.
Losing muscular control and that hydrostatic balance they rely on makes it hard for them to move, to feed, to survive.
Then what happens?
As it progresses, distinctive white lesions emerge on the surface. These are patches where the tissue starts to decay, looking pale, necrotic, standing out against their normal color.
Okay, white spots.
And these lesions are followed by progressive decay of the surrounding tissue. They spread rapidly, the body's integrity just visibly breaks down. These lesions can advance to literally bore holes right through the epidermis, the skin, and into the underlying muscles.
Eating away at them.
Essentially eating away at the animal from the outside in. It's a literal disintegration while it's still alive.
Oh, that's horrible.
Internally, the animal becomes limp. That's due to the failure of its water vascular system, that unique hydraulic system sea stars use with their tube feet for moving, feeding, maintaining balance. It collapses internally.
So they can't move their little tube feet.
Can't extend or control them, movement becomes impossible. And a hallmark of advanced SSWD is widespread epidermal ulceration. The outer skin just breaks down completely, exposing tissues. And limb autotomy.
Autotomy, they drop their arms.
Yes. Sometimes they purposefully detach arms, maybe in a desperate attempt to shed infected tissue. Other times the arms simply fall off because the tissue integrity degrades so much they just can't stay attached anymore. While autotomy can be a normal defense for sea stars, or even asexual reproduction in some, here it signifies severe pathological distress, a desperate, often futile sign of breakdown.
So they go from healthy to...
They transition dramatically. From a healthy state, puffy arms sticking straight out, good turgor, to visibly sick and deflated. Arms limp, curled, maybe detached. The whole body looks shrunken, discolored.
And how fast does this happen?
Remarkably rapid, frequently leading to death within just a matter of days, or at most a few weeks from the first signs.
Days. That's incredibly fast.
Incredibly fast. The internal structure continues to break down, culminating in complete body disintegration. It's often graphically described as melting. The sea star literally liquefies into this pile of white mush on the seafloor.
Melting. Good grief.
It's a truly horrifying sight, and that rapid visible decay, it wasn't just alarming, it played a huge role in grabbing widespread public and scientific attention. It was so unlike anything normally seen. You just don't expect animals to melt in nature.
No, you really don't.
And a critical, awful aspect of this progression is that the decay itself fuels the problem. As infected sea stars disintegrate, they release vast amounts of organic material into the environment.
Food for bacteria.
Exactly. It paradoxically fuels the proliferation of the very bacterial communities responsible for the syndrome. This creates a powerful positive feedback loop. Dying individuals feed the microbes causing the disease, accelerating its spread and severity within the population.
That's a vicious cycle.
A truly vicious cycle.
So for over a decade after this huge 2013 outbreak, the actual cause remained a mystery. That must have been incredibly frustrating for scientists.
Immensely frustrating. A true scientific whodunit with enormous ecological stakes. Early research, especially studies published around 2014, they initially and mistakenly implicated a virus, a densovirus, specifically the sea star-associated densovirus, or SADV.
A virus seemed plausible, I guess.
It was a reasonable hypothesis at the time. Many epidemics in other animals are viral. However, subsequent, more rigorous investigations conclusively demonstrated that SADV was likely just a common normal resident in healthy sea star populations. It wasn't actually causing the wasting.
So a false lead. Correlation, not causation.
A classic example of correlation not equaling causation. A pitfall you often encounter in complex systems. It was there, but it wasn't the killer.
What made it so hard to find the real culprit?
Well, a key methodological hurdle in those early studies was relying heavily on tissue samples from dead or dying sea stars.
Okay.
Those samples often lacked the crucial coalomic fluid. That's the internal body fluid, kind of like their blood or lymph. That's where the active disease agent would likely be most concentrated during the acute phase.
So they were looking in the wrong place, or maybe too late?
A bit of both, perhaps. Plus, just tracing environmental diseases, especially underwater in that complex, vast marine environment, it's recognized as immensely difficult. The scale, the water movement, the microscopic nature of pathogens. It's hard to pin down a source or vector.
So how did they finally crack it?
The pivotal breakthrough came only recently, with research published in the journal Nature, Ecology, and Evolution. It definitively identified a specific bacterium as the culprit behind SSWD, Vibrio pectinacida.
A Vibrio bacterium. We mentioned Vibrios earlier as potentially persistent in the ocean.
Exactly. This particular Vibrio species is also known to infect shellfish, which is an interesting clue about potential cross-species interactions or shared environmental reservoirs.
And how did they pin it on this Vibrio? What was different this time?
The key was a refined methodological approach. Scientists meticulously analyzed the sea star's coelomic fluid, the internal fluid, rather than focusing only on external tissues where countless other environmental microbes live and could easily confuse the results.
Focusing on the internal fluid was the key.
That shift in focus proved critical. And then they did rigorous lab experiments to prove causality, really trying to nail down those Koch's postulates.
What kind of experiments?
First, they injected coelomic fluid from 6 sea stars into healthy ones. And guess what? The healthy ones developed the wasting symptoms. That confirmed a transmissible agent was in the fluid.
Okay, step one.
Then, crucially, they heat-treated the fluid, first killed any living microbes, and then injected it. When they did that, it prevented disease development. That confirmed it was a living, heat-sensitive agent, like a bacterium or virus, not a stable toxin or something else.
Okay, getting closer.
And finally, they successfully isolated the Vibrio pectinacida strain from 6 stars, grew it in a pure culture in the lab, and then showed that administering that pure culture to healthy sunflower stars induced the illness.
That sounds like it hits all the marks.
It satisfies the crucial parts of Koch's postulates, providing that definitive link, finally, after a decade.
So how does this bacterium actually cause the wasting? Do they know the mechanism?
The proposed mechanism involves the Vibrio pectinacida proliferating, growing rapidly, on the sea star's surface and potentially within its tissues. As the bacteria multiply, they consume oxygen, potentially forming biofilms, and this leads to a reduction in oxygen availability right at the sea star's tissue surface.
So the sea star basically suffocates.
Effectively, yes. It's thought to cause respiratory distress at the cellular level. And this process is then exacerbated by the decay of infected individuals, providing more organic material for the bacteria to grow, closing that awful positive feedback loop we talked about.
Wow. That whole scientific journey, the false leads, the persistence, finally getting the answer, really shows how tough science can be, especially in the ocean.
Absolutely. It's a testament to the scientific process, iterative, challenging, but ultimately finding answers.
So what does this breakthrough mean now for tackling SSWS?
It's huge, but there's another critical piece to this puzzle.
Oh, what's that?
While Vibrio pectinacida is now identified as the direct cause, scientists widely acknowledge and strongly suspect that warming ocean waters act as a critical contributing factor.
The climate change link.
Exactly. Amplifying the disease's impact and severity. This isn't just a coincidence. It seems to be a profound synergistic interaction. Climate change is like a major predisposing factor, making the disease much worse.
How does warmer water make it worse?
Well, elevated water temperatures impose significant physiological stress on marine organisms, especially cold-blooded ones like sea stars. It's like running a constant low-grade fever for them.
Stresses their bodies.
Makes them more susceptible to disease in general, increases their vulnerability to microbial infections, even from common environmental bacteria like this Vibrio.
Does it affect their immune system?
Yes, the stress can also directly impair or reduce the immune function of the sea stars. It diminishes their ability to effectively fight off pathogens.
So they're stressed and their defenses are down.
Exactly, and we have evidence for this. A smaller outbreak on Vancouver Island back in 2008 already showed that higher water temperatures directly facilitated the syndrome's spread and increased mortality, an early warning sign.
Right.
Further research shows a clear correlation between water temperature and mortality rates during the big outbreak. Warmer waters accelerated the disease's progression, it killed them faster, and increased the likelihood of succumbing to the illness at all.
So temperature is like an accelerator pedal for the disease.
That's a good way to think of it. In fact, the relationship between temperature and where sunflower sea stars are found actually changed significantly after the SSWD outbreak.
How so?
It suggests that cooler habitats may now be acting as vital refuges for the species. Areas that maybe stayed a bit cooler were where the survivors persisted, implying that historically suitable warmer areas might now be too dangerous for them.
Wow. And historically, have these wasting events been linked to warm water before?
Yes, past, smaller SSWS outbreaks often correlated with El Niño events. those periods of anomalously warm ocean water across the Pacific. So there's a long-standing historical link between heat and outbreaks, just maybe not on this massive scale.
Okay. But the big 2013 event wasn't initially linked to warm water.
The initial emergence wasn't broadly linked to elevated temperatures right at that moment, but subsequent prolonged warm water conditions, like that infamous marine heat wave called the blob that persisted for years afterwards, are believed to have significantly exacerbated the outbreak's severity and its incredibly wide impact.
So the heat wave poured fuel on the fire.
Exactly. Consequently, these massive outbreaks are increasingly viewed as a direct product of climate change. Rising global temperatures are enhancing the vulnerability of marine life and facilitating the transmission and severity of diseases like this.
So it's not just the bacterium.
It's the bacterium supercharged by a changing environment.
Which means the problem of sea star wasting syndrome isn't just a simple pathogen-host thing. It's this broader, much more complex crisis, sitting right at the intersection of marine health and global climate change.
Precisely.
And that means tackling SSWS isn't just about fighting the bacteria, it's somehow about the temperature of the entire ocean, which that adds a whole new daunting layer of complexity to finding solutions.
It absolutely does. It links this specific ecological disaster directly to global climate pattern.
Okay, let's talk about the consequences.
Yeah.
The scale of the SSWS outbreak was unprecedented, you said, stretching across vast distances, killing billions.
Yeah.
Just how far did it reach, and did it hit all sea star species equally hard, or were some tougher than others?
The 2013-2014 outbreak showed this absolutely unprecedented geographic reach, impacting sea star populations, as we said, along the entire Pacific coast of North America, from Baja all the way up to Alaska.
How did it spread so fast?
It rapidly disseminated from that initial observation point in Washington state, indicating really efficient spread mechanisms, maybe through water currents carrying the Vibrio, or maybe through the movement of infected animals before they died, or maybe a combination.
And was it just the Pacific?
Well, the primary and most severe impacts were definitely on the Pacific coast. But SSWD has also been responsible for sea star die-offs on the Atlantic coast, particularly in New England.
The Atlantic, too. Same disease.
It indicates either the pathogen has a much broader distribution than initially thought, or perhaps multiple related occurrences of similar wasting syndromes are being triggered by similar environmental stressors, like warming waters on both coasts. It adds another layer of complexity to understanding its global potential.
And was it picky about which sea stars it hit? Or did it affect lots of different kinds?
Crucially, the wasting disease is not host-specific. It affected a diverse array of over 20 sea star species.
Over 20. Wow.
Yeah, that wide host range makes it particularly damaging ecologically because it impacts multiple different parts of the food web simultaneously.
Were some hit worse than others, though?
Definitely. The sunflower sea star, Pycnopodia helianthoides, that big keystone predator, was disproportionately impacted. It suffered the most severe declines, with populations plummeting by roughly 90% across its range, basically gone in many areas.
90%. That's why it's endangered now.
Exactly. That catastrophic decline led to it being formally declared critically endangered in 2020 by the IUCN, and the U.S. government proposed listing it as a threatened species in 2023 under the Endangered Species Act. It highlights just how severe the impact was, demanding urgent conservation action.
What about the ochre star? The common intertidal one.
The ochre star, Pisaster ocrasis, another really crucial keystone species, was also profoundly affected. Population declines of 85–90% in places like Oregon, and an unbelievable 99% or more at over half the surveyed sites down in Southern California.
99%.
Yeah.
Just wiped out.
Essentially functionally extinct in large parts of its southern range. Other affected species included mottled stars, leather stars, though, interestingly, some leather star populations seemed unaffected, pointing to potential regional resilience. Six-armed stars, rainbow stars, giant pink stars, giant stars, sun stars, vermilion stars, bat stars. A whole list.
But some species were okay.
Yes. Conversely, some species, like Henricia levisiscula, the blood star, appeared to be largely unaffected. They maintained their presence even in heavily impacted areas.
Why is that variation important?
This observed variation in susceptibility is absolutely crucial. It strongly suggests there are underlying biological differences between species, maybe differences in their immune responses, maybe genetic predispositions to the pathogen, or maybe varying ecological interactions that protected some.
So studying the survivors is key.
Exactly. Studying these resistant species, or even genetically distinct surviving individuals within the heavily impacted species like Pycnopodia or Pisaster, can provide invaluable insights into the mechanisms of natural resilience. Why did they survive when others didn't?
And that knowledge could help conservation.
Immensely. It's paramount for informing and developing targeted conservation strategies. If we can figure out what makes some resistant, maybe we can leverage that through selective breeding, perhaps, or specific habitat management to help bolster the vulnerable populations. It's like a natural experiment playing out, giving us critical clues.
Okay, so a keystone species like the sea star gets massively removed from the ecosystem. You mentioned they eat urchins. What happened when the sea stars disappeared? What were the ripple effects?
The ripple effects were dramatic and immediate. The mass die-off of sea stars, especially the sunflower sea star, directly precipitated an unchecked increase and explosion, really in the numbers of sea urchins across affected regions.
The urchins went crazy.
Their populations exploded. Without their main predators, these proliferating sea urchins, which are voracious grazers, subsequently just mowed down extensive kelp forests, leading to widespread habitat destruction.
How bad was the kelp loss?
They effectively graze the kelp right down to the rock, consuming the holdfasts that anchor the kelp. They leave behind what scientists call urchin barrens, vast areas stripped totally bare of kelp, transforming these vibrant three-dimensional forest habitats into desolate, rocky seafloors dominated only by urchins.
Like a desert underwater.
An underwater desert, exactly. A striking example is Northern California, where kelp coverage plummeted by an astonishing 95% within a single decade following the SSWS outbreak.
95% of the kelp forest gone.
Gone. A direct, devastating consequence of this ecological imbalance. The removal of the sea star predator, allowing the urchin grazer to run rampant. It's a classic trophic cascade.
And losing kelp forests is a huge deal, right? You call them the rainforests of the ocean.
A massive deal. Kelp forests are incredibly vital marine habitats. That rainforests of the ocean analogy is used because of their exceptional biodiversity and their critical importance. They provide shelter and food for a huge array of marine mammals, fish, invertebrates.
A whole ecosystem relies on them.
An entire complex ecosystem. They create structure, like a terrestrial forest. They're nurseries for juvenile fish, foraging grounds for sea otters, protective cover for countless invertebrates. So the disappearance of sea stars, leading to the loss of kelp, has had significant ripple effects through the entire food web.
Affecting fish, otters?
Impacting fish species that live in the kelp, crabs, lobsters, even larger predators like sea otters and seals whose prey base diminishes or disappears. It ultimately reduces overall marine biodiversity. It compromises the functional integrity of the ecosystem, making it less resilient to other shocks.
Like future heat waves or pollution.
Exactly. And it significantly diminishes the ecosystem services that these rich habitats provide, things like carbon sequestration, nutrient cycling, coastal protection. The whole system becomes less robust, less productive, less stable without its keystone predators keeping that balance.
So this wasting syndrome, was it totally new in 2013, or had it happened before?
That's an important point. SSWS is not a completely novel phenomenon. Historical records indicate it has been around for decades. There were smaller-scale die-off events documented back in the 1970s, 1980s, and 1990s.
Oh, okay.
And often, those earlier, smaller events were associated with warm water El Niño events, which again suggests that recurring pattern linked to environmental fluctuations, or warmer waters might have historically triggered more localized outbreaks.
But 2013 was different.
Unequivocally different. The 2013-2014 outbreak was unprecedented in its scale, its geographic scope, its speed, and the sheer magnitude of mortality. It dwarfed anything seen before.
And has it gone away now? Or is it still lingering?
It has not disappeared. That's another critical point. It has persisted at low levels in most affected areas and continues to cause sea star mortality. It often seems to flare up seasonally.
Seasonally, like in warmer months.
Potentially linked to temperature fluctuations, yeah. And maybe also linked to the sea star's reproductive stage, which can be physiologically stressful and might temporarily lower their immune response, making them more vulnerable at certain times of the year.
So it's shifted from a huge epidemic to something chronic.
Exactly. It suggests a fundamental shift from a series of isolated, episodic outbreaks to a more chronic, endemic threat The disease is just kind of always there now at some level.
Why would it become endemic like that?
It implies that the marine environment itself, likely influenced heavily by climate change-induced warming, has become more consistently conducive to the pathogen's activity, or perhaps just maintains the sea stars in a state of heightened vulnerability. The baseline conditions have shifted.
And this constant pressure,
Mhm.
could it drive evolution?
It could. This continuous disease pressure might be driving rapid evolutionary adaptation in the surviving individuals, selectively favoring those with higher natural resistance. We could potentially see a more resilient, albeit perhaps a genetically altered, population emerge over time, but that takes time, and the populations are still severely depleted.
Right. And you mentioned the impact wasn't the same everywhere. Southern California got hit harder.
Yes, significant geographical disparity. Southern California experienced much more severe declines, often over 99% loss compared to northern regions like Alaska or British Columbia, where declines were still bad, but maybe not quite as catastrophic.
And the North might recover faster.
Interestingly, yes, northern regions are predicted to potentially recover faster, maybe due to higher juvenile recruitment rates from healthier source populations further north, and perhaps the benefit of consistently cooler, less stressful waters.
So this uneven impact is useful scientifically.
It offers invaluable natural experiment opportunities for marine disease ecologists. They can compare the factors genetic, environmental, ecological that might confer resilience in the north versus those that exacerbated susceptibility in the south. By studying these differences in depth, they can directly inform more targeted conservation strategies, identify which populations might be most robust, which need the most help, and what conditions promote recovery.
Okay, so given this dire situation, a keystone species critically endangered, entire ecosystems transformed, the disease now endemic, what are scientists and conservationists actually doing to fight back, to help sea stars recover? Is there any hope?
There is definitely hope and a lot of dedicated effort underway. The recent identification of Vibrio pectinacida as the primary cause was a huge step. It marks a pivotal moment because it finally allows for more targeted interventions.
Right, knowing the enemy.
Exactly. Before, they were effectively fighting in the dark. Now, ongoing studies are focused on understanding exactly how this Vibrio spreads in the wild, whether it's native or introduced, what other environmental factors beyond just temperature might influence future outbreaks.
Looking for other triggers or amplifiers.
Right. And a really promising avenue is the potential development of probiotics.
Probiotics, like yogurt for sea stars.
Kind of. The idea is to help healthy sea stars resist the disease by bolstering their natural microbiome with beneficial microbes. Lab studies are exploring the microbe communities found on healthy surviving sea stars, and even looking at using phages.
Phages, viruses that kill bacteria.
Exactly. Viruses that specifically infect and kill bacteria, like Vibrio pectinacida. So, exploring if beneficial microbes or specific phages could be used to increase sea star survival, perhaps administered before reintroduction efforts. It's exploring biological control options.
That sounds fascinating. What about broader conservation strategies?
They're being developed through really multifaceted approaches, often coordinated by large groups like the Nature Conservancy, working with government agencies, universities, aquariums, tribal partners. It's a big collaborative effort, outlined in things like the Roadmap to Recovery for the Sunflower Sea Star.
Okay, what are some key parts of that roadmap?
Well, a major component is captive breeding and potential relocation.
Breeding them in labs or aquariums?
Yes. Scientists are exploring testing the remaining healthy wild sea stars to identify individuals with natural resilience to the disease. Then, establishing carefully managed captive breeding programs to increase their numbers.
And then putting them back in the wild.
That's the long-term goal. Relocating healthy captive-bred individuals, or resilient wild ones, back into decimated regions to try and jumpstart population recovery. This involves scaling up the number of facilities that can do full life cycle captive rearing, ensuring genetic diversity is maintained.
How do they maintain genetic diversity?
Through things like cryobanking.
Freezing samples.
Exactly. Expanding cryobanking involves collecting and cataloging reproductive samples, sperm, and eggs from across the species' remaining range. These frozen samples can be used later to introduce genetic diversity into the breeding programs, preventing inbreeding and preserving the existing genetic variation for the future. It's like a genetic insurance policy.
Smart. What else?
Disease challenge trials in the lab. actively exposing different family lines to the pathogen under controlled conditions to identify which ones are inherently more resistant and what genetic traits confer that resilience.
Finding the toughest families.
Finding the tough ones, yeah. And continuing to explore those biotic interventions like probiotics or phages that could potentially be given to sea stars before they're outplanted to give them a fighting chance.
And monitoring is key, I assume.
Absolutely crucial. Long-term monitoring programs are essential to track wild populations, density, recruitment, size structure, distribution, growth rates, and the characteristics of their habitat. Plus, evaluating the genetic structure of wild populations to understand connectivity and local adaptation. All this data needs robust management to integrate findings across different regions and groups.
Are they setting specific recovery goals?
Yes, efforts are underway to develop clear demographic and genetic recovery goals.
Yeah.
What does recovered actually look like for these species? And this includes assessing other threats beyond just SSWD. Things like accidental bycatch in fisheries or habitat impacts from pollution or coastal development. The recovery plan has to be holistic.
Right. Can't just focus on the disease if other things are hurting them too.
Exactly. And before any relocations happen, there are comprehensive disease risk assessments done. You have to be incredibly careful not to accidentally spread other diseases when moving animals around. Research is also testing various outplanting techniques to see what works best in different habitats and establishing monitoring for how well the released animals do.
Sounds like a massive coordination effort.
It is. Addressing all the permitting challenges, ensuring biosecurity in captive facilities, and engaging the public. It all requires really transparent communication and collaboration among dozens of agencies, partners, communities. Public outreach is vital to build support for these long-term, complex efforts.
But there's still challenges, right? Funding.
Huge challenges persist. The future of critical research facilities, like the USGS Marrowstone Marine Field Station, which does a lot of this vital work, faces uncertainty due to proposed budget cuts.
That would be terrible timing.
Devastating. Such cuts could severely jeopardize the ongoing research needed to understand and combat marine diseases, setting back recovery efforts significantly. And just the inherent complexity of tracing environmental disease sources, especially underwater, remains a massive hurdle. It makes proactive management incredibly difficult and costly.
So connecting this back, these huge concerted efforts, they really highlight how critical both the cutting-edge science and this massive collaborative action are for trying to save not just a species, but entire marine ecosystems.
Absolutely. It's a complex, multi-layered problem demanding a multi-layered, long-term solution. It shows human ingenuity and commitment, but it's also a very stark reminder of the immense scale of the challenge we face in our changing oceans. Hashtag, tag, tag, outro peril.
Well, as we resurface from this deep dive into the hidden epidemics of our oceans, it's just abundantly clear, isn't it? Marine diseases aren't just some fascinating scientific curiosity.
No, they're much more than that.
They are a profound and often really alarming indicator of our planet's overall health. We've seen how these underwater epidemics, like fibropapillomatosis in sea turtles, clearly driven by human environmental cofactors,
The pollution, the nutrient runoff.
and the catastrophic sea star wasting syndrome, amplified dramatically by warming oceans. They represent these intricate challenges that are just fundamentally different from what we typically deal with on land.
Precisely. These cases powerfully underscore that the health of marine organisms, from the tiniest bacterium up to the biggest sea star population, is intimately linked to the health of the entire coastal ecosystem.
And that reflects back on us.
It directly reflects the impact of human activities, often on a global scale. That One Health approach we talked about. It offers such a crucial, really indispensable lens through which to understand and hopefully address these interconnected crises. It reminds us we are all part of the same vast living system.
What affects the ocean eventually affects us.
It has to.
So the journey we've traced from scientific mystery surrounding these diseases to finally identifying causes, from devastating outbreaks to these multifaceted, complex conservation efforts, it reveals the incredible inherent resilience of nature sometimes.
It does. Nature fights back.
But also its immense and often surprising vulnerability in the face of rapid change.
Yeah, especially change driven by us.
So, as you, our listeners, reflect on this journey through the hidden epidemics of our oceans, what new connections stand out to you? How might a deeper understanding of these seemingly distant marine diseases maybe inform your own approach to health, to environmental stewardship, maybe even just in your own backyards or communities or daily choices?
It's worth thinking about that interconnectedness.
It really is. It's all part of one vast living system, and the health of the ocean truly is, in so many ways, our health. This has been the Deep Dive.
Thanks for diving in with us.
We hope you feel a little more well-informed and perhaps a little more connected to the vital pulse of our blue planet.